immunology. You'll also see as I'm doing the immunology, everything I've been doing thus far, I did a lot of art therapy for you guys. This one, there isn't much art therapy. I found videos. I won't show the videos in here, but they'll be recorded. They'll be in the recordings. I'll verbalize everything, and I'll show you over here. I do a lot of my analogies too, putting things together. All right, so this is immunology. These words, this is probably how you're going to feel after going through immunology, and the antibodies going around his head, B cells, T cells, NK cells. They'll all make sense to you. Some people like to do flashcards for this because it's just easier about this is what the cell is, this is what the cell does, put it on the other side. All right, so let's do this. All right, immune system, fighting against something. Now, how I do this is explain to you as though you're protecting your house. And we have three forms of defense, three lines of defenses to help protect your house from burglars. First off, we have the first line of defense. And what that is in my situation, that's a picket fence around your house. Okay? That's telling someone on the street that this is private property. Now, people could come in and out of there, can break it. It's not very strong, but it's not bad. I mean, you see a picket fence, most people won't break through it. But they can break through it. These are things like your skin, all right? Secretions from your skin, mucous membranes. You know, you have bacteria that goes into your nose. It's going to be sequestered by the mucus over there. But it can, if there's enough there or strong enough, it'll pass right through the mucous membranes. Or it'll pass through your skin, you know what I'm saying? But it's there, it's protecting friend and foe. It's protecting you from friend and foe. In other words, there's a mailman, there's Antilles, there's a burglar. That picket fence is telling everybody that this is private property. Does that make sense? That's our first line of defense, and it protects you from friends or foe. Then we have a second line of defense. The first line of defense and the second line of defense are what we call non-specific. In other words, it's not going to ward off friends. It's not going to ward off enemies. Both of them will come through here, right? Antilles and a burglar is going to go through the picket fence. It's not going to protect it from both. Our second line of defense, if someone breaks through that picket fence, our second line of defense is Fido, the guard dog. So he's sleeping, someone breaks through the picket fence, what is the guard dog going to do? Start barking. No matter if it's the postman, which is a good guy or a good girl, right? Or a burglar, that dog is going to bark no matter what. Friend or foe. So because of that, the picket fence and the guard dog are what we consider non-specific. It's telling us that something has invaded us, whether good or bad. Does that make sense? Okay? Now, we got to be more specific, though, because the postman's okay to come in. But burglar's not. That dog barking is going to alert the master that's inside the house. What's all that barking for? Fido, what's going on? Now, that master is very specific. He knows friend and foe. Fido, that's Antilles, that's okay. You can lick her leg, it's okay. The postman, that's okay. He can come in, and it's fine. A burglar is something different. Fido, keep barking. Grab his foot. I'm going to call up 911. You see, the master is very specific. He or she knows if that is friend or foe. And when it's a foe, meaning an enemy, the next thing that he's going to do is to talk and get backup over there. The policeman, the squad car. Well, he doesn't know what to get. He just knows to get 911. Bless you. And the dispatcher on 911 is going to be the communication between the master, the the house it's invaded, and everything else. The dispatcher is probably the most important part of the whole immune system, because that dispatcher knows, okay, you have a fire, we're going to get the fire department there. Or you got a bomb, we're going to get the bomb squad there. Oh, someone fell down, we're going to get the ambulance there. That dispatcher knows exactly what to bring. It's very specific. So can you visualize what's going on here? Okay, these two, as much as it looks pretty busy, this is actually easier to deal with. This has so many parts. Even though it's just showing lymphocytes and antibodies, there's a lot of parts to this. This is where we get a little bit more crazy. And it's the connection between the master and the dispatcher connecting with whatever things need to be over there, that gets really involved. So think of it like that. Don't think of it as I don't want to say challenge. Don't think of it as like a chore. Think of it as something fun. Use these analogies to help you along. I find that students have a harder time with this also because it's hard to visualize it. It's like chemistry. Chemistry makes it, you know, to see, let's say, a water molecule, and you got to deal with hydrogen bond. You have to visual, because you can't see it. It's not here in front of you. A heart is easy to deal with because you have a heart in front of you. You can understand, oh, that's what a chamber looks like and stuff. This is hard. This is more abstract. Does that make sense? So you're going to have to, my way of studying this kind of stuff when I went through it was to come up with these stupid cartoons in your head to make you see it more visually. And that'll help you along with it. And just build your story getting bigger and bigger and bigger. And don't worry about if your idea is making sense to you, but when you explain it to someone else, it does, because everyone has their own experiences, and whatever works for you might not work with someone else. So don't let that say, like, well, am I thinking it differently? It's working, it's working. Okay? Questions about how we're going to go on to this? Okay, so immunology is broken down into what we call the non-specific, which I just explained to you, all right, the picket fence and the guard dog. This is what we call the innate immunity system, all right? Innate means non-specific. And they're going to be things like your physical barriers, like your skin. It's going to be cellular, and we'll talk about some cells that'll do this, the chemical barriers. If food goes down, or bacteria goes down to your stomach, you got acid down there. That acid's going to kill off that bacteria. But it's also going to kill off certain other good things too. So it's non-specific. Okay? And then we're going to talk about a whole slew of what we call soluble mediators. Chemicals that get released from other things to talk with other aspects, to connect things. So this is our non-specific, i.e. the picket fence and the guard dog. But then we have the more specific thing that we refer to as the acquired immune system. And that's broken down into two things. Something we call humoral, and it's not humorous like this, or ha-ha-ha, funny humorous, but humoral means antibody related. Okay? We have B cells that are going to make and synthesize these antibodies. And we'll explain what antibodies do. Then we have a cellular branch to this acquired immune system, and it's going to be cell-mediated. And it's going to tell cells what to do. The main component of that is going to be T cells. Now, why do they call them B cells and T cells? Because these were originally, what were these originally, B cells and T cells? We talked about it earlier. A certain type of white blood cells. No, lymphocytes. Lymphocytes become B cells or T cells. When lymphocytes go into the bone marrow, they're going to be matured into B cells. Bone marrow, B cells. When they get into the thymus, thymosins, those hormones, mature those lymphocytes to become T cells. Thymus, T cells. Okay? So these are the two acquired or adaptive cells that are going to be controlling the whole system on this specific side. Okay? Like I said, I'm going very slow with this. Eventually you'll be able to pick up with this stuff, but there's a lot of definitions we got to deal with. And how I usually deal with this is, there's two ways to teach it. You either show the scrimmage, show the football game, and then introduce all the characters, or do you show the characters and then show the scrimmage or the football game? I choose to define everything first and then show you the scrimmage. It just makes, for me, it makes a little bit more sense. So, here's our non-specific system and all its players. All the football players in the non-specific. Very busy. We'll get through them all. Some of them, if you look at it, some of them you could already recognize because we dealt with those before. Eosinophils, basophils, and so forth. The acquired immune, or the more specific immunity, deals with antibodies and the B and T cells. And as I said to you before, even though you have a lot more over here, this is much easier to manage than what we got to deal with over here. This is the picket fence and guard dog. This is the master and the connection he or she has with the dispatcher and who to get there. This gets busy. Now, how does your body know something's foreign? Well, if it's something from the same individual, we say it's autologous. Meaning, I'm going to burn my nose, and now my nose is all grinded down. Look like a skeleton. So they're going to take a piece of my buttock, my skin over here, and they're going to make a nose out of it, and they put it over here. It's not something I want to smell all day long, but will my body sense that that is foreign? No, because it came from my own body. That's what we call a graft. Okay, and you've probably seen those or heard about those. Some of them, they're also called flaps if they're going to do the same area. But that your body doesn't see as foreign, because it has some sort of antigen it already recognizes. This is a situation where we're going to get a kidney from your own identical twin. Syngenous. And we're going to get into that too. This is pretty much autologous because it won't fight against it, and we'll explain that. But if it's coming from an identical twin, that's very good. But we see a lot more of this happening. Allogenous. This is found within the same species. I need a kidney. I don't have an identical twin, so I'm going to have to ask my next door neighbor if there's a match. What is that match all about? We'll get into. But that's what we see a lot more of. Will your body fight against that? Yes. Okay? So if you have an identical twin, don't ever get into a fight with him and her. You might need his or her kidney. Okay? And then we also have the synonymous. And this is actually getting something from another species. This is where you hear about the pig heart, or the baboon heart, or the valve that came from a pig and put it into a human. Again, you would expect your body to fight against this also. So we have ways that we're going to have to decrease your immune system so that your body doesn't fight against it. But you could imagine that if you decrease your immune system, that means you're a good opportunity for other bacteria to go against you, and now you can't fight it as well. So it's a double-edged sword. You've got to weigh the pros and the cons. Now, we could also deal with the size of the antigen, because if the size is so small, your body can't even recognize it as foreign. Okay? A mouse going through that picket fence could get right through there, probably could pass right through the dog, and even the master probably won't see the mouse. It's so small. So if it's less than what we call 5 kilodaltons, it's not immunogenic. It's too small for your body to recognize whether friend or foe. But an antigen, when it's attached to something, then we could actually, it's big enough where your body can see it. And that's called a hapten. We take that mouse and we attach a long red flag to it. The mouse is still small, but now we can recognize the mouse and the flag. A dog's going to see it, might have some hard time to get into the fence, but sure as heck the master will see it too. If it's less than that, it won't be immunogenic. So these are just certain things, and I won't ask you about these, but these are just certain things that are immunogenic and things that are not immunogenic. It's about the size. Okay? So again, I won't ask you about this, it's just there to be complete. Now let's look at an overview of the innate system. All right? That's the non-specific. These are things that we'd be talking about. Barriers, the picket fence, your skin, the fluids in your eyes, the chemicals in your stomach. We also have something called the complement system, which we'll get into. It's going to release certain things to amplify other things. We also have interferons, which we'll talk about too. Phagocytes, certain white blood cells and certain other cells are going to engulf the bad people. And we also have something called inflammation, which is also going to help fight things. But again, these are all non-specific things. Whether friend or foe, these things are going to affect that. So the exterior defenses, skin, cilia, mucus, okay, these things are going to try and get that up there. Whether you have sawdust in your nose, or you have bacteria in your nose, or you have a finger up your nose, the cilia is going to try and fight that. You're going to irritate it enough where there's going to be mucus coming out of your nose. Okay? We also have commensal microorganisms in certain areas. We've got good bacteria and bad bacteria in the vagina and in the mouth. The good bacteria, this commensal organism, microorganisms, is going to keep the bad bacteria down. They may fight for the same food. Okay? I'll give you an example of where this is into play. A lot of women like to douche. And douching is cleansing the vagina with vinegar, or they have these chemicals that you can use, and it's fine. All right? A lot of women like to do that just before they want to see the gynecologist. Very similar to also you want to gargle with hydrogen peroxide in your mouth. Sure, it'll make your mouth clear of killing off bacteria. But here's the problem with douching and hydrogen peroxide in the mouth. It's going to destroy not just the bad bacteria, but also the good bacteria. When you destroy the good bacteria, the bad bacteria sense that, and they actually flourish more than the good bacteria. So they start building up, having an opportunity to start growing and causing mayhem. It's not just bacteria, also fungus. That's why women who douche a lot actually will build up more of a yeast infection, because the yeast starts building up inside there. Because the good bacteria usually homes down on the good the fungi, which is the yeast infection. They might get oral thrush, which is a yeast infection also. Okay? So usually what I tell women, it's fine. You want to do it, it's fine. Don't do it often. You want to do it once a month, just after your period. If you need, for yourself, if that's what you want to do, that's fine. But I had some women that want to do it like every other day. That's way too much. Once a month is fair if that's what you want to do. And don't do it before you see the doctor. That's the other issue, because it actually, when they do a pap smear, it disrupts that, and you're going to have to go for another pap smear. So it kind of ruins the results of that. So, but some women prefer to do it, and that's fine. Just don't do it often. Okay? So that's a little bit about commensal organisms, microorganisms. So again, exterior defenses, and then we also have biochemical defenses, things like the acid that's in your stomach. It's going to kill certain bacteria. There are certain bacteria that will love that low pH. Certain bacteria you'll learn in microbiology called Helicobacter pylori, or H. pylori. That one loves to be in such a low pH, and actually, because of that, it causes mayhem and causes stomach ulcers. But for the most part, a lot of things don't like that low pH. We'll learn about the hydrochloric acid that's in the stomach is at a very low pH. It's like 2. You put a drop of hydrochloric acid on your hand, it will come out the other end over here. Right? It's that strong. And there's some other things that are also in here that are also the exterior defenses. But again, this is all non-specific stuff to defend you against friend or foe. Okay? So here's some other ones, more specific now. We have phagocytic cells. We have phagocytic cells, cells that want to engulf something, bring it in, and break it apart, digest it. We have fixed phagocytes, meaning phagocytes that stay, not in the bloodstream, but are fixed in a certain organ. We call these macrophages. And they come from the bone marrow. Where from the bone marrow? Well, what were they once? Macrophages were once what in the bloodstream? What type of white blood cell? Monocytes, right? They're the largest white blood cells. Monocytes are like little police officers. They're patrolling the streets. But now the commissioner says, you know what? We're doing a wonderful job on the streets, but in the malls, the crime rate is going up. So I don't want you to be patrolling the streets anymore. I don't want you in the bloodstream as a monocyte. Now I want you to be stationed at the mall in the liver. And you're going to be fixed there. You don't even need to come onto the streets. You're going straight over to the, and so you're going to be living in there. Now you're not a monocyte anymore on the streets. You're a macrophage that lives in the liver. All right? Monocytes can engulf, and macrophages can engulf. It's just where they're located. Okay? Now, if you're located in the liver, you're called a Kupffer cell. So a macrophage in the liver is called a Kupffer cell. A macrophage in the intestines is called Peyer's patches. A macrophage in the kidney is a mesenchymal cell. A macrophage in the brain is a microglia cell, and so forth. They're all macrophages. They just have special names of where they're located. Okay, questions about that? All right. Then we have the circulating monocytes, which I explained to you about. Those are the patrolling police officers that are going all over the bloodstream. Okay. And neutrophils. Neutrophils also are phagocytic. They have granules as much as monocytes do, but these specific granules contain either lysozymes or lactoferrins. Lysozymes are split cell walls of many gram-positive bacteria. Now, I'm not going to go into, I'm assuming you guys haven't taken microbiology. Who here, just a show of hands, who here has taken microbiology?
You can't unless you guys
Right, but I mean prior to today. No, you could actually take microbiology and AMP1 at the same time at this school.
Oh.
Yeah.
Didn't even realize that. I wouldn't
Okay.
know that.
That's fine. That's fine. You don't need to. You don't need to take microbiology for what I'm going to teach over here. And I'm not going to go into the details of what gram-positive and gram-negative bacteria, but this is just to give you an idea of what it is, very briefly. All the bacteria that's out there, and there's tons, thousands, and millions of bacteria, can be broken down into gram-positive and gram-negative bacteria. It depends on the type of cell wall it has. Okay? And it stains differently. Purple for gram positive and pink for gram negative. But you don't need to worry about that. But I'll be popping up gram positive, gram negative up there. Just know it as bacteria for you. But for people who took microbiology, you'll see the bridge there. Okay? So lysozymes are as a chemical that's going to break up the cell wall of many what we call gram-positive bacteria, certain bacteria. Okay? Lactoferrin is a chemical that binds to iron, and many bacteria need that iron for their living. So this actually takes away that, and then they can't survive and they start dying down. So that's what happens with some of these granules that are actually in the neutrophils to help fight off certain bacteria. It breaks them down. Okay? So how does phagocytosis occur? What's the mechanism of it? I'm going to explain it over here, but the picture will make more sense of it. But basically, well, actually, let me just show you the picture. It'll be easier. Okay? And I'll get back to what an opsin is. Phagocytosis is, this is a bacteria, and this is a macrophage. And the macrophage is going to engulf that bacteria and bring it inside of it. Did you see that? Let me show you again. Okay? Macrophage sees the bacteria, it starts engulfing it, brings it inside, and then it's going to break up that bacteria. Okay? Macrophages are pretty good. They can do more than one at a time. Sometimes when they get overwhelmed, they got to call in other things. They got to call, the dog can bark away and bite and kill that burglar and stuff. When it gets overwhelmed, it's got to have a master come out, and we got to get the specific system coming in. So this is what happens here. This is the macrophage. That little blue thing up there is going to be a bacteria. The macrophage is going to engulf that. So now you have this bacteria that's inside of a vesicle inside the macrophage. And it's going to fuse with something called a lysosome. Right? We learned this from cytology. A lysosome is filled with digestive enzymes. That lysosome will fuse with this, the phagocytic vesicle. And now you have this combination called a phagolysosome. Those enzymes go in, break up that whole bacteria, and destroy it. And then it goes out, whatever, the back end or whatever. And you have a lot of this going on. That's what pus is. Okay? Garbage. But one other thing that is not showing on here that's very important for you to understand is that it doesn't throw out everything. It keeps a piece for itself as a trophy. It keeps a piece of it and acts as an antigen and will stick out later on so that the acquired immune system sees it and recognizes it and knows what to do. We'll get back to that. But it's going to stick out a piece of it. Most of it will go out, but a piece will stay behind, and we're going to utilize that. One other thing I want to just add over here is this bottom line here. Opsinin. You're going to see that quite often in immunology. Opsinin is anything that's going to amplify or enhance phagocytosis. Okay? Whether it's special glue or another chemical, whatever it is, it's going to enhance phagocytosis. Okay? We'll be using opsonization, which is the process of using opsin. We'll use that quite often. Again, we're still with the non-specific. Natural killer cells, NK cells. These are also lymphocytes too, but these are not specific lymphocytes. These are non-specific. And these will specifically kill cancer cells and virus-infected cells, not bacteria. They're going to be stimulated by a group of chemicals called soluble mediators. We'll get into those later. They will also release something called perforins. Perforins, as the name implies, it's going to perforate or punch holes. It's going to drill holes in the cell membranes that have been infected with cancer cells or have been infected with virus cells. Put holes in there. Now, if you put holes into a cell, then the fluid around it has salts. It's going to go in there, it's going to disrupt the membrane, and the action potential gets disrupted, the whole cell will burst. And I'll show you that later on. So natural killer cells are a certain type of non-specific lymphocytes. They secrete potent chemicals to aid in what we're going to get into with inflammation. There's no prior exposure that needs to be set on here. It will affect everything, whether good or bad. You don't need any memory to that. They don't mature in the thymus. There's no surface antigen receptors on these guys, and these are found in the spleen, blood, bone marrow, and LN stands for lymph nodes. So that's where they'll be located. We also have some accessory cells, non-specific. We have mast cells. These are basically basophils that you learned from before, but basophils float in the bloodstream. These stay in organs. Now, they are not derived from basophils as much as macrophages derived from monocytes. Mast cells are not made from basophils, but they kind of are analogous with a macrophage is like a mast cell in the organ, whereas a basophil is like a monocyte in the bloodstream. The only thing is that monocytes make macrophages, basophils do not make mast cells. Does that make sense? Okay? So these will be found in the CT's connective tissue of organs, and they release histamine. We talked about histamine. It's going to vasodilate blood flow, or blood vessels, to allow more blood to go into those areas. It's also going to allow fluids. If, for instance, you have, let's say, mast cells in your nose. You have an allergen that comes in, like pollen or something. It comes in. Mast cells are going to release histamine. It's going to vasodilate the blood vessels. So now you have more blood flow there. That's why your nose appears very red, because it comes closer to the surface of your skin. Vasodilation happens, more blood flow there, more white blood cells. Red blood cells go there also, because there's a lot of activity that's going to happen there. They need a lot of oxygen too, so the red blood cells are going to bring that there. You got more white blood cells going there to help fight the infection. They also become very permeable, or the blood vessels become very permeable, allowing the white blood cells to squeeze on out of there with less resistance. Problem is, because they open up, become more permeable, the blood vessels become more permeable, they allow the white blood cells there, also fluid goes out there also. It's going to follow. And that's why you start talking like this, right? Because those are so stuffy. So what do you give yourself so that you'll get a stuffy dose? Antigen. It'll bring down the effect of histamine. Heparin is going to be there, so it's going to prevent less clotting. Okay, you don't want many clots to happen over there, so that's what's going on. You have a platelet activator factor, eosinophil chemotactic. We'll get into these later on, but it also releases those things. And there, basophil. So like I said, basophils and mast cells, they basically do the same thing. All right, it's just the mast cells are located in the cells themselves, or in the tissues themselves. Okay? And you learned about basophils from before, so I'm not going to bore you. Then you also have eosinophils, right? Learn about those? Now, eosinophils, they also do phagocytosis. They increase when we have parasitic worms, okay? And they specifically will surround the parasitic worms, engulf it, and break it up, and I showed you all that, what it does. It's a piece of it later on. It also is going to release chemicals to inhibit histamine. So when you have an allergy, we're going to increase eosinophils. We're going to also increase basophils and mast cells. But we don't want the mast cells and basophils to get too hyperactive, because then you're just going to be miserable, right? You're going to have a nose that's so stuffy, you can't talk, can't breathe. So the eosinophils come in and kind of tone it down. So they increase in terms of allergies, but not to fight off the allergies, but to make sure that your immune system does not become hypersensitive. Does that make sense? So you could bet that there's a lot of studies on this. When people have allergies, they might have something wrong with their eosinophils, because the eosinophils might not be suppressing this so well. Right? Okay, an antigen-presenting cell. Remember we were talking about when something engulfs something, everything it's all broken up and it goes out. They leave a piece of it, a piece of antigen for something else. That's where this is coming in. An antigen-presenting cell. Okay? I'm a macrophage. I've just engulfed a certain bacteria. Goes out my poop scoop. But I have a piece of it sticking out here. I'm an antigen-presenting cell, which is an antigen-presenting cell. There's a whole other antigen-presenting cells, but a macrophage is one example of one. I'm presenting that antigen to someone else. Okay? Just the way it sounds. Antigen-presenting cell takes up the antigen and presents it to the acquired immune system. The dog barking is presenting the fact that someone just broke through and presenting it to the master. Does that make sense? It's presenting it. These are the types, these are the antigen-presenting cells we have. We have things called Langerhans cells that present to virgin T cells that have never seen that antigen before. We have dendritic cells that also present to virgin T cells. We have B cells that present to T cells that have seen that before. And then we have our favorite macrophages that also present to T cells. You can see how busy this is getting with these words and stuff. Looks mean, but this is what's happening. You have this antigen-presenting cell, let's call it a macrophage, who just went through phagocytosis and broke up all that, but a piece of it was left and was able to have it stick out over here. So now this is presenting this antigen to this mean guy. This mean guy is good. He's on our side. It looks like a villain. But we need some bad guys, not bad guys, but like some, heavy-duty guys, right? Harley Davidson, like tough guys. That's what we need to help fight wars and stuff. So that's what we have over here. He doesn't know what to do. He doesn't know who the enemy is. But this guy is presenting. If you have anything that looks like this, or at least the antigen that looks like this on anything, I want you to destroy it. You're giving him knowledge. This is the non-specific. This is specific. He now knows what to recognize and destroy because of the help of the non-specific system. Does that make sense? Okay? Now, there's a whole slew of chemicals that come out of these things, these cells. And maybe flashcards might work, a chart, whatever works best for you. But there's a whole slew of them, so I want to go over some of them with you right now. They're chemicals, soluble mediators. One is called C-reactive protein, or yes, they do call it crap. Okay? And C-reactive protein, it binds to certain microorganisms and act as non-specific opsonins. They kind of act, they get attached to these bacteria, and that makes it like a certain glue where if something's going to phagocytize, it's going to be able to bring it in much better. And opsinin is going to enhance phagocytosis, and that's what that is. Now, if you have a lot of C-reactive proteins in your bloodstream, that's telling us that we have some infection that just happened recently. We call it an acute infection. Not cute like adorable, but acute. All right? Some doctors and clinicians, for that matter, like to use that as a marker to say, look, we have no idea what you have, but your C-reactive protein is very high. That's telling us you have some acute infection going on with you. I don't usually, I'm the 50% that doesn't really use it. Some doctors and nurse practitioners and other clinicians like to use it to give them some clue what's going on. It doesn't help me per se. If someone's got a fever, I could figure out if they have an infection or not. I have other means that I can do that, which is way over the scope of this course. So to me, for me, that's a wasted marker. I don't need to spend the $50 on it. Whereas some people, it's going to help them. They have no idea what this person has. Let's start someplace. Hey, the C-reactive protein is low, so therefore we know there's no acute infection. So there could be a chronic infection. You know what I'm saying? It doesn't tell you if you have an infection or not. It just tells you if you had an infection that, for the past couple of days. Okay? So some people, that's why when you get into your clinicals, some doctors may want the C-reactive protein, some people might not. But that's what that's used for. Okay? We have a bunch of cytokines. Cytokines are small proteins that get secreted by a certain signal to tell it to get secreted. We have cytokines that get released from macrophages, and those are called monocrines. Makes sense. Okay? Or monokines. If they get released from T cells or B cells, they're ones lymphocytes, so we call them lymphokines. Okay? We also have two types of cytokines. All right? And we dealt with this before, and this is more or less an anatomical categorization. Cytokines that get released from macrophages are monokines. Cytokines that get released from T cells or B cells are called lymphokines. But we also talk about their physiology. Do they actually act on itself? Then it's an autocrine. Or does it act on a different cell that's next to it? Then it's a paracrine. And we talked about autocrine and paracrine with endocrinology. So that's where that comes in too. Okay? Complement system. My, that's a nice quote. No, wrong complement. Okay? Complement system is a cascade of events that occurs where we're going to have two different pathways. All right? And we'll get into those later. There's two different pathways, almost like extrinsic and intrinsic pathways when we deal with the clotting system. We have something called the classical and alternative pathways, which we won't go into right now. But the thing is, there is 5 functions of the complement system. One of them is going to enhance phagocytosis, so it's going to do opsonization, act as an opsonin. It's also going to create an inflammatory response, so it's going to increase mast cell degradation, and also with that, in that sync, we're going to also increase capillary permeability. It's also going to lyse things. It's going to destroy and punch holes in bacterial cell walls and destroy them. When you punch holes there, fluid comes in, it changes the concentration inside and outside, and the whole membrane gets disrupted and destroys it. It also acts as a chemotaxis. Chemotaxis is kind of like, if I want people to follow me, I'm gonna leave like a popcorn trail, and they're going to follow where the popcorn goes, and they're going to see where I am. So chemotaxis is releasing chemicals so that other things can follow where it is, right? Chemotaxis. Okay. And then we're going to eliminate any of the immune complexes. All right. So it's going to clean up the whole thing.